Good morning. Right out there. Good morning. Good morning. There we go. I'm Dr. Tim Smith, the Associate Provost at the college, and I'm happy to welcome you today to our Provost Forum talk featuring Dr. Melanie Styers. Before we get started, though, two service announcements, two favors I would ask of you, please. First, your phones, either turn them off completely or put them on vibrate at least. And then for those who are sitting towards the back, if you turn around, we are videotaping. We always videotape these. We have found that if you're sitting right close to the camera and you talk, we don't hear the speaker on the video. We hear what you're saying, what you had for lunch, what you're doing later today, how hard that math exam was. If we would ask you please not to talk if you're going to sit right close to the camera. It's now my distinct pleasure to introduce today's speaker. Dr. Styers is an Associate Professor of Biology who joined the BSc faculty in 2010. She holds a BS in Chemistry and Mathematics from Birmingham Southern College and a PhD in Biochemistry, Cell and Developmental Biology from Emory University. She's an active researcher, active grant writer, and I guess grant getter as well, right? If that's such a thing. The title of her talk today is very timely. I think many of us can relate to it. In Sickness and in Health, Novel Roles for Membrane Trafficking Proteins in Disease. Please help me welcome Dr. Melanie Styers.
So, thank you for coming out today. Does everybody hear me? Am I pretty loud? Yes? Good to go. Okay, well, thank you for coming today, and what I'm going to be telling you about is some of the work that I did on my sabbatical, and today I'm really going to be telling you about two stories, both related to human diseases that were published in the last year. So one of these stories is going to use a human disease to inform some of the work that my lab did in Fission Yeast, which is the model system that we used. And the second story is going to be where our work in Fission Yeast informed some work that we did in human cells in collaboration with a laboratory at UAB. So I'd like to start by first saying thank you. So thank you to the Provost Office and the Faculty Development Committee for supporting the sabbatical program, as well as my department, and Issa and AJ, all of the people who really helped to make research happen at Birmingham Southern College. I would also like to thank UAB. So my sabbatical was actually in the laboratory of Elizabeth School in the Department of Cell and Integrative Biology at UAB. And in particular, I worked very closely with Justina, Yunju, and Akil on the project, on the work that I'm going to be talking about today. I'd also like to thank all of the members of the Styers Lab, starting all the way. Matt Barnes was my first research student ever in 2010. So sometimes it takes a really long time to get a story complete, but all of these Birmingham Southern students contributed to the story that I'm going to be talking about today. And I'd like to thank the NSF, who also supported this work as well. And finally, I think it's really important that we as researchers, particularly I think sometimes as women engaged in research, it can't be done without having a very supportive partner. So I'd like to thank my husband, Justin Robertson, who I think is coming today but isn't quite here yet, as well as my daughter, Anna Catherine, who deals with mommy being in and out and carrying her to the lab and everywhere else. So I'd like to thank them as well. So I want to start, I want to start by showing you something. So you're probably thinking, what do I care about membrane trafficking and what is it? So I'd like to show you why I find this topic so exciting. So this is a video of a viral protein as it moves through the cell over time. So what you can see here is that this protein moves really rapidly. It's found in these tiny dots that look like they're moving with a purpose towards the center. Then you see more of these tiny dots that look like they're moving with, so they kind of collect in the center, and then they start shooting outward from the center out to the edges of the cell. And here, what's going to happen is that this protein is actually moving to the cell surface. But hopefully what you can appreciate is that this is a really highly organized and highly orchestrated process that occurs within the cell. And in reality, we still don't understand much of how this occurs at the cellular level. So my lab is really interested in understanding the cellular mechanisms that allow these processes to happen. How is movement of proteins throughout the cell organized, and what kind of machinery does it take to make that occur? So as I mentioned, the protein that I just showed you is a viral protein. And one reason why understanding these processes is very important is that many viruses co-opt these intracellular membrane trafficking systems in order for their own replication. That means that if we can understand how these systems work, then we can target these systems to develop new antivirals. But that's actually not the story that I'm going to be telling you today. So today, I'm going to be talking about two other diseases. So the first is Chilaiditi syndrome. This is a lysosomal storage disease, a pigment dilution syndrome, that it's a genetic disorder that children are born with. So I'm going to tell you about that story and how our work has informed our understanding of CHS. And then I'm also going to tell you a completely new story. In my work in the stool lab, we were able to identify a new role for membrane trafficking proteins in glioblastoma. So glioblastoma is a really devastating kind of brain tumor that is essentially incurable. So at our current state. So our work is providing new insight into how glioblastoma occurs. So I want to start by thinking, in order to understand how membrane trafficking works in sickness, first we have to understand something about how it works in a healthy individual. But I want to start by drawing an analogy to help you understand. So imagine that the United States is like a cell. So the United States has lots of different regions that has lots of different, each region has its own defining characteristics, right? So each region has sort of a local flavor. And I think many times we define that local flavor in food. So for example, I hail from North Alabama. Is anyone else in here from Decatur? No one? Huntsville?
Oh yeah.
Okay. So if you're from North Alabama, then you're probably familiar with our white barbecue sauce, right? That's a North Alabama thing. So we are known for that local flavor. So what local flavor is this? New York, right? Oh, this is the flat pizza, not the thick pizza. It's the flat kind. So New York. What about this one? Chicago, right? Chicago dogs. This one was for the Provost. If you're not from the Midwest, you may not get this one. Does anybody know what this is? It's a hot dish. I have a good friend who's from Iowa. It's this weird casserole that they make that always has tater tots. And then Spam. Where does that? Where do lots of people eat Spam? Hawaii, right? That was for Joseph. So each region of the country has these local flavors that are associated with these characteristics that we automatically associate. But there has to be some way to move between these regions. So we have lots of transportation mechanisms where we could vary all of the ingredients that it takes to make these local flavors. So for example, you could take a car, right? That's one mechanism of transport to go from Alabama to New York. And this is for my preschooler. You could also take a train to move from New York to Chicago. You could take a magic school bus to head to Iowa. And you would definitely need a plane to go to Hawaii, right? But each one of those mechanisms of transport is very different from one another. So my goal as a scientist is to understand two things. So in the cell, what are these local flavors? What are the characteristics of these different places within the cell, and how do we move things from one place to another? So now I'd like to tell you about some of those places within the cell. So first, one of our organelles is the endoplasmic reticulum. The endoplasmic reticulum is studded with ribosomes, and that's where lots of membrane proteins and secreted proteins are synthesized and gotten ready for transport to other places. Then we have the Golgi apparatus. So it's this stack of pancakes looking organelle in the middle. This is a major hub for sorting, and it's also where proteins get modified. So these purple pieces attached to this protein here, those are all sugars. And these sugars get trimmed and modified, and additions are added to them in the Golgi apparatus. We also have the plasma membrane, so the cell surface. It's important for lots of different reasons: how cells talk to one another, how different tissues communicate, but also how cells move. So cells can form these lamellipodia to move from one place to another due to changes in the plasma membrane. And then an organelle that's near and dear to my heart is the lysosome. So this is like the recycling center of the cell. So anything that is old and used up that needs to be degraded can be broken down into its component pieces, so those component pieces can then be recycled. And as I said, we don't just care about the local character of these organelles, of these parts of the cell. We also care about how things move between them. And how things move between different places in the cell are in these small membrane sacs called vesicles. And there are different types of vesicles that shuttle materials between different organelles. So those are our transport systems, our Thomas the Train, our Magic School Bus. Okay, so just to remind you, the key questions that I'm interested in addressing are: how do organelles maintain that local flavor? And what I'm going to be talking to you about today is, in particular, what proteins are important for maintaining the homeostasis, sort of the stable environment of the lysosome, that recycling center of the cell. And then how do proteins road trip between these different organelles? How do we move proteins between them? And this is where I'm going to be talking about proteins that make vesicles and their function in glioblastoma. So I'm going to start with this story. So I want to start by talking about this Chiari-Kagashi syndrome, or CHS. So this is a CHS patient, and you may notice here that this child's hair and eyes are very light. That is characteristic of CHS, because the organelles that are affected are lysosomes and organelles that are related to the lysosomes. Well, the melanosomes are the organelles that give pigment to our hair and skin, and those are a lysosome-related organelle. So that leads to partial albinism. Another organelle that's affected are the platelet granules. So platelets, as you know, are involved in clotting, so there are clotting defects that occur. And here you can see that the lysosomes themselves look different. So wild type here, this is someone who doesn't have the disease. But in these patient cells, this is stained for the lysosomes. You can see that they're much larger. They look like big bubbles. So the lysosomes are enlarged in these patient cells and non-functional, or not as functional. So how this manifests as far as clinical outcomes, the biggest danger to these patients is that they're immunocompromised. So let's talk a little bit about why that occurs. So this is a type of white blood cell called a neutrophil. And the role of neutrophils is to take in pathogens like bacteria and then essentially kill them. So it kills them in several ways. They can spit out toxic substances at them, but they can also take them and form these phagosomes that then fuse with the lysosomes. And as you remember, the lysosome is the recycling center of the cell. So we can kill these pathogens and degrade them so that they can't harm the organism anymore. So when that happens, the neutrophil will then die, and it's taken up by another white blood cell called a macrophage. So that's the normal situation. But what happens in these patients is those bacteria, shown in yellow here, are still taken up into the neutrophil. However, they're not killed. And instead, the pathogen will often lyse the neutrophil and then escape, allowing the bacterium to proliferate. So this leads to immunosuppression. These patients are immunocompromised. They can't deal with those bacteria. So luckily, a lot of research has been done on this disease in humans, and we know exactly what gene is mutated and what causes CHS. And it's a human gene called LYST or LYST. And this gene it creates a really big protein that's associated with the lysosome in human cells. And we know something about the function of this protein as well, because it has partner proteins that are present on the phagosome. So this is where your bacteria would be taken up. So we want these two organelles to fuse so we can then kill that microorganism. So these two proteins come together, and we think that what LYST does is it serves as a scaffold to bring together a lot of other proteins in order to drive fusion of these two organelles. And depending on what tissue you're considering, these proteins have been shown to be involved in both fission and fusion of these organelles, so it seems to matter what those partner proteins are and what they're doing. But the major question is, we really don't know what the partners are. We have no idea. Under what situations LYST interacts with other proteins and what those proteins are, which are involved in fission, which are involved in fusion. And so that's where my lab comes in. We were interested in exploring this question using yeast as a model to understand what these other proteins may be. So now let me tell you a little bit about yeast. So this is the fission yeast, Schizosaccharomyces pombe. So this is a yeast that it's shaped kind of like a pill, and as it grows, it gets longer and longer, and then it divides by medial fission, meaning it separates in the middle. So these yeast have they share many of the same genes that we have as humans, and that means that we can look at genes in yeast to help us understand what may be going on in human cells. The other thing that's nice about yeast is that you can easily delete genes and look to see what the effects are. So the homolog, the related protein to LYST, is LBS1 in fission yeast. So if LYST and LBS1 had the same function, remember that patient cells have these really big lysosomes. So we would expect that yeast that have LYST deleted, that have LBS1 deleted, would have really big vacuoles. Because in yeast, the equivalent organelle to the lysosome is the vacuole. Still serves as that recycling center, has much of the same function. And hopefully what you can appreciate here, so this is our normal cell, it has all of its genes intact. But when we delete LBS1, those vacuoles get much larger. So the other interesting thing that we found is in my advanced cell biology class, a group of students who were doing an individual research project as a part of the class happened to find another gene that also affected vacuolar size. So deletion of a gene called YPT4 resulted in vacuoles that were even larger, and in fact there was even an effect on cell size. The vacuoles are really large and the cells are really large as well. And so you'll believe what I'm saying, we also quantified these results. So we measured the biggest vacuole in each cell, and you can kind of imagine this as blue would be small vacuoles, red would be medium, green would be large, and then purple would be extra large. So you can see wild type mostly has medium-sized vacuoles. YPT4 and LBS1, however, when you remove those genes, they have large and even some extra large vacuoles. So the vacuoles are much bigger in this situation. So this suggests that our model does in fact work, that the yeast we're seeing the same outcomes as what we see in patient cells. So we wanted to explore this further since we knew that yeast lacking these genes have enlarged vacuoles. And we wanted to try to consider how these cells work together, or how these genes work together. So here you can see, these are the wild type cells. Once again, this is pretty much very similar data to what I just showed you. Whereas in YPT4 and in LBS1, we're getting more of those large and extra large vacuoles. So to see if these cells were working together, we wanted to see if these genes were working together. We wanted to see if we give cells lacking LBS1, if we give those cells extra YPT4, will it fix the problem? And if we give cells lacking YPT4 extra LBS1, will it fix the problem? So we started by giving cells extra YPT4 to see if it fixed it. And so just as proof of principle, this is a good thing. If you give cells YPT4 that were deleted for YPT4, so you give it back what it's missing, it largely fixes the problem. So this, you lose those extra large vacuoles, and it starts to look much more like normal cells. However, if you give cells lacking LBS1, if you give it YPT4, it doesn't fix the problem, and in fact, it may even make it worse. So that seems strange. So then we did the reverse experiment. We gave cells that lacked YPT4 LBS1 to see what would happen. So one thing that we noticed was when we added too much LBS1, the vacuoles got smaller, even in wild type cells. Similarly, if you gave LBS1 back its own LBS1, the vacuoles got much smaller. You can see that we have mostly small and medium vacuoles. However, more importantly, if you gave cells lacking YPT4, if you gave it back LBS1, it fixed the problem in those cells. So the vacuoles became smaller again. So what this says is that overexpression of LBS1 fixes the problems with vacuolar size in cells lacking YPT4. So now this suggests that these two genes may be working together somehow in a pathway. So we wanted to understand that a little bit better. So I told you that these proteins are sometimes thought to be involved in fusion, sometimes thought to be involved in fission. And you can get big vacuoles in two ways, right? You can have too much fusion or not enough fission. Fission meaning the vacuoles dividing. So we did an experiment to test if fusion or fission were impaired. And so yeast is really convenient. If you subject them to osmotic stress, you just place them in water with no salt. They will fuse their vacuoles. And you can see here that in the normal cells, and especially in YPT4 and LBS1, those vacuoles get really, really big. So this suggests that fusion is okay, and more importantly, that we weren't at some maximum fused state already. But then, if you take them out of that water mix and put them back in their normal media, then those fission will occur. Those vacuoles will go back to their normal size. And what you can see is that here in the YPT4 and LBS1, they didn't really seem to go back to their normal size, even though their normal size was already bigger. So these vacuoles were even bigger than what they were in their original state. So this suggests, although it's not proof positive, that fission may actually be what's impaired in these cells. But probably more important, we wanted to understand, are these enlarged vacuoles affected? Because in patient cells, remember I told you that the lysosomes are functionally impaired, so they can't degrade those bacteria that are coming in. So to understand this, you have to understand something about the function of the yeast vacuole. So first, the yeast vacuole is very acidic. So lots of protons are pumped into the vacuole, so it's a very acidic environment. This is also a place where lots of ions are stored, for example, calcium. So because of this, yeast that have impaired vacuolar function are sensitive to conditions where the media is more neutral or basic. They're happier in an acidic place. And they also become sensitive to high levels of calcium because they can't sequester it away and put it in that vacuole. So we wanted to test to see if these functions were impaired in our mutant cells. So first, just to give you an idea what this is. So this is called a spot assay. And these are our normal cells. I want you to look here first. So this is a 10-fold dilution. So as you move to the right, each spot has 10-fold less cells. So you can see that when we put these cells on their normal Yeast media, they grow fine, they're really happy. And in fact, when we put wild-type cells on neutral media at pH 7, they're even happier. And then, even though they're not quite as happy, these wild-type cells, when we grow them in the presence of calcium chloride, they can still grow pretty well. So now if we compare that to our mutant cells, so our YPT4 cells and our LBS1 cells, cells lacking these genes, you can see that they grow fine on a normal media. They're doing fine. However, you see many fewer cells either in the presence of neutral media or in the presence of calcium chloride. So this told us that the vacuoles were in fact functionally impaired, helping us understand that both of these genes do also affect the function of the vacuole. So loss of either one of these genes resulted in sensitivity to neutral pH or calcium chloride. So this led to a model. So in yeast, high salt, changes in nutrition, all of these types of changes in the environment can affect vacuolar size. So our model is that essentially YPT4 can sense these changes in the environment, in the local environment, such as what I showed you earlier where we had changes in osmotic pressure. And YPT4, we think, then activates LBS1 to form that scaffold and recruit whatever those unknown proteins are that then drive vacuolar fission. So one interesting thing here is we were really looking for these proteins, right? We still don't really know the identities of these proteins that bind LBS1 and drive that fusion. It seems like YPT4 is probably acting upstream of LBS1. But the really important part of this story is that there is a human version of this YPT4 protein. It's called RAB4. And we have no idea if it plays a similar role in human cells. So this is sort of the nature of my work, is we learned something about human cells, use that to inform our work in yeast, and now we can go back to human cells to see if that holds. And maybe we can put a new protein into this disease pathway in humans. Okay, so that's sort of we're halfway through, sort of my first story. So I'm going to completely change directions now and talk about a different story that I also worked on during this time. And that story is about those vesicles. Remember, we talked about that there have to be transporters that move proteins between these different organelles. So I want to tell you a little bit about the proteins that form those vesicles, that form those transporters, and then I'm going to tell you about our new findings about how these proteins function in the context of glioblastoma. Okay, so just as a reminder, we're talking about moving proteins between all of these different places within the cell, and it's these vesicle carriers that are moving things back and forth. So to understand that, we need to think a little bit about how these vesicles form. So this would be an example of, this is our donor compartment. So let's say that maybe this is the endoplasmic reticulum. So we have a protein that needs to move from the endoplasmic reticulum, and we're going to send it to the Golgi. So that's our target compartment where we're going to move it. So these proteins are often found either associated with the membrane or inside this organelle. So in order to form that vesicle carrier, the membrane has to bud outward. So it actually starts protruding and forms this bud that then pinches off. So scission occurs right here. And then this vesicle is carried along the cytoskeleton, which my cell and molecular biology students know about. So it's carried along the cytoskeleton and then delivered to its target compartment where it fuses. You can almost imagine this as when two soap bubbles fuse. That's what's occurring here. So the two soap bubbles are separating and then fusing. And when that fusion happens, then the contents can be dumped into this target organelle. So now I need to introduce you to a group of proteins that my lab spends most of their time studying. And these are the GBFs. So GBFs are enzymes, which are protein catalysts, so they catalyze biochemical reactions. And essentially, these are enzymes that activate other proteins. So GBFs stand for guanine nucleotide exchange factor. But what that means in practice is how they activate these proteins is by causing them to bind a molecule, a nucleotide called GTP. So the GBFs that my lab studies activate these proteins called ARFs. And so when that GBF encounters that inactive ARF, it's bound to this low energy GDP molecule. The GBF causes it to kick off this GDP, and then it can bind GTP, which is a high energy form of the molecule, allowing it to become active. So now we have an active ARF, and it can do its work to form the vesicle. Now this process can also be reversed by another enzyme called GAPs, and GAPs essentially remove part of this molecule to put it back into its inactive state. So this is a never-ending cycle of activation and inactivation. So now I want to help you understand why that matters. And I think the easiest way to understand this is through a cartoon. So here, we're back to our donor membrane and our target membrane. So once again, this would be like our endoplasmic reticulum. We're going to form a vesicle that then goes to this acceptor membrane. So this would be like our Golgi apparatus. So first, remember we have this inactive ARF, so it's bound to GDP. It's in its low energy inactive state. When it encounters a GBF, it's going to activate it. And it activates it by causing it to bind GTP. So now our ARF is active, ready to go, ready to make a vesicle. So how it does that is it brings in its friends. So the ARF recruits these coat proteins, and coat proteins are almost like the ticket takers of membrane trafficking. Because essentially what they do, if this were the magic school bus, the coat proteins would look at the passengers, decide if they have a ticket, and then let them onto the bus. So this is a cargo molecule that wants to make it all the way over here to this acceptor protein. So these coat proteins start to help curve the membrane and form this vesicle, which is then going to pinch off of the membrane. We're going to move this vesicle along the cytoskeleton, and then it eventually fuses with its target membrane where the contents can be released. But just to go back to the very beginning, what started the entire process was those GBFs. So the GBFs have to be there to activate the ARFs or nothing can happen. So as I mentioned, my lab is interested in understanding what these GBFs are and how they function. And we already know, I told you that there are different types of transporters that move to different places. So there are different GBFs and different types of transport vesicles that move between each of these different organelles. And here they're sort of symbolized by these blue, red, and yellow color-coded vesicles or budding vesicles. The ones that I'm going to be talking about today are the ones shown in red. And these are the guests that we're interested in here are guests of the GBF1 Gaia family. So GBF1 is the human name for this protein, and Gaia1 is the yeast name for this protein, which my lab has spent some time characterizing. And it's also important to note that this is the family of proteins that has been linked to the Ebola virus, the hepatitis virus. It's required for replication of these viruses. So understanding its function is really important from a developing antiviral standpoint, in addition to the story that I'm really telling you. So we've already learned some things about these proteins in my lab. So my lab published a story a few years ago trying to understand the function of Gaia1, this family of proteins in yeast. And what we learned is that yeast that lack one copy of this Gaia1 gene build these. It's a structure called a septum. So when the cell is about to divide, remember it divides by splitting in the middle. This is what it should look like. However, this is what it looks like in cells that have mutated Gaia1. Sometimes you get multiple septa, sometimes the septa are splitting in the middle, and sometimes they're just weird. I mean, that's crazy, right? Nowhere near the middle. So we know that loss of Gaia1 alters septum formation. And one thing that's really key to understanding why this is important is because fission yeast are a pretty unique model because they grow in a really directional way. So when a new fission yeast cell is born, essentially first it grows in one direction, then it switches direction and takes off and starts growing in the other direction. Then it forms this septum right in the middle, and then this septum has to be degraded to understand in order to separate into the two new cells. So what that means in practice is that there has to be a polarized, a directional transport of the proteins that are going to get rid of, that are going to degrade this septum. And my lab did a lot of work to understand this in the past, and what we found is that one of these proteins that degrade the septum, ENG1, shown here, so normally it should be found at the septum where essentially it acts like Pac-Man and degrades it so the two cells can separate. What we found is that in our mutant cells that didn't have Gaia1, it's never sent to the septum. So we knew that polarized transport was messed up in these cells lacking this family of proteins. So this is a case where my lab's previous yeast work is going to inform the work I did in human cells. So what we wanted to know, since we knew that polarized directional transport was messed up in yeast cells, we wanted to see if this polarized transport was messed up in human cells. And so it's important to understand not all human cells are polarized. Some cells don't really have much of a direction. Some cells do. So for example, neurons, right? They have dendrites and axons, and those are on different ends. So there are directions that you can move towards. Well, another type of polarized cell is a migrating cell. So a cell that's moving from one place to another. And we knew that one place where migrating cells are really important in a clinical way is in glioblastoma. And this is showing you what that looks like. So here, these U87 cells are glioblastoma cells. And this once was a monolayer, so a solid layer of cells. And someone has put a scratch down the middle of those cells. And over time, you see that those glioblastoma cells crawl right back in and fill in that scratch. So these cells are really, really highly migratory. They move in a direction. And as I mentioned, this is a polarized process because these cells will extend this lamellipodium and then grab the substratum with these focal contacts in order to move in a direction. So we wanted to see if the human version of Gaia1, GBF1, was involved in that process, that directional movement. So the first thing we did, if it was involved in this process, the first thing we wanted to do is look and see where Gaia1 is in these cells. But before I get there, one quick note about why this is important. Glioblastomas arise from these star-shaped astrocytes that begin growing out of control. The problem with glioblastomas grow rapidly. They're very invasive. They invade the surrounding tissue, and it can cause a lot of pressure in the brain, as you can imagine, and the symptoms vary widely, all the way from headaches, nausea, and it also can cause changes in speech, memory loss, and behavior. The treatment largely in glioblastoma is resection to remove the tumor. However, because these are so invasive, it's almost impossible to get the whole tumor, so recurrence occurs frequently. So understanding how these cells migrate may help to block that invasive behavior where it invades the rest of the brain. So this is a non-polarized human cell, these HeLa cells. And here you can see, when we look at GBF1, so this is the human version of GBF1, it's in this little ring around the nucleus. This is the Golgi. So that's where it localizes in a non-polarized cell. But now we wanted to look in these glioblastoma cells to see where it was, just to start. So what we found, surprisingly, is in glioblastoma cells, we see it in the Golgi, this blob next to the nucleus, but it's also present in the tips of those cells where they're sending out these processes thinking about migrating. So this was completely novel. This protein had never been seen at the cell surface ever. So we wanted to try to figure out, well, if it's at the tips, where is it? So we knew it was present at the leading edges of these processes. So we wanted to consider what kinds of other proteins might also be there. So to understand that, I told you these cells also make these focal contacts. And this is how the cells sort of attach to the substratum and drag themselves along. So cells are almost like slugs, sort of. So they attach and then drag, attach and then drag themselves, push themselves forward. So they use these contacts called focal adhesions to do this. So this would be inside the cell, and then this is the road that they're attaching to in order to move themselves forward. And these are really well characterized, and there are some known proteins that localize to these focal adhesions. And I'm pointing out two examples here, alpha-actinin and vinculin, because they're going to be in the next slide. So what we asked next is, well, is GBF1 in the same place as these focal adhesion proteins? And the answer is yes. So here you can see GBF1, so shown in green here, so we can still see it at the tips of these cells, indicated by the arrows. And shown in red, alpha-actinin and vinculin are also in these same places. And just to make sure you believe me that it's in the same places, we can overlay these images, and in microscopy, red and green make yellow. So you can see the yellow staining, suggesting that GBF1 is present at these focal adhesions. So, in science, questions just lead to more questions. So we were actually expecting to try to figure out what GBF1 was sending out from the Golgi in a polarized manner. Well, now we have a completely different question, because GBF1 is actually where we expected it to be sending things. So our question is, what is GBF1 doing here? Is it making vesicles? Is it doing that function that I showed you earlier that we know it does? If so, what's in these vesicles? So who are the passengers? And we also wanted to know, how does GBF1 get there? We didn't expect it to be there, and we have no idea how it got there. So I did start answering this question briefly, and this part is especially for my biochemistry students. So we wanted to know if the structure of GBF1 contributed to its membrane recruitment. So here you can see one part of GBF1, it has this small piece called the HDS3 domain. It's just a piece, it's a conserved piece of this protein. We had this idea that GBF1 may have a closed state where the HDS1, the door is shut, or it may also have this open state where the door opens and then maybe it can bind to membranes when it's in this open state. So in order to start testing that, we had to purify the protein, and this is what I'm teaching in class right now. This is very timely. So my goal was to try to purify this HDS3 domain. So this small piece of GBF1. Well, purification can be a really difficult process just to purify it out of human cells. So instead, I tried to grow up this piece in bacteria. But to do that, I attached a tag. So this tag is called GST, and it lets us pretty easily purify the protein. So the first thing I wanted to do is see if was this protein actually expressed in the bacteria. So this is my control. If I just have GST by itself, it is expressed when I treat the bacteria to get them to start expressing the cell, or to expressing the protein. And then in addition, we can also see a little bit of this new band, and this is my GST protein right here, my GST HDS3. So this was good. I could express it, but now I have to get it out of the cells. So in order to do that, GST is really convenient because it binds this molecule called glutathione. So I could just use these glutathione beads to essentially pull that GST out of solution. So I could attach it to these glutathione beads and see if I could purify it. So the next question is, could I purify it? Well, that worked too. So this is GST by itself, and here's my GST HDS3. So now I know I can purify this protein using what technique? Anybody in biochemistry? Good job, affinity chromatography. So I purified this protein using affinity chromatography. But remember, the question was, is this HDS3 going to attach to GBF1? Because if so, that suggests that it can be this door that puts it in the closed state. And here are my results from that experiment. That's not a mistake. Here are my results from that experiment. This was about 3 months of my life. So essentially, I got no interpretable results, and we still don't know the answer to this question. But I think this was actually a really good experience for me because it put me back in that state of understanding where my students are when they're in the lab doing research. So I think something that I got back in touch with really nicely is that sometimes science is hard, sometimes it doesn't work the way you expect, and that's okay. That's how life works. You just have to keep trying at it. And I think that was really good for me to reconnect with that feeling that I know sometimes students in my research lab experience. So in addition, finding the answers to questions also raises lots and lots of new questions. And I told you that I never got the answer. So there's never enough time on a sabbatical or really any time to complete all the things that you want to finish. So for those of you who are taking notes in order to turn in your extra credit, so the two key take-home messages from the story that I've told you today are that YPT4 and LBS1 both play key roles in maintaining vacuolar size and function in yeast, and that may be really important for helping us to understand what's happening in that disease, Chidachi-Gashi syndrome, in the future. And second, that we have discovered a completely new localization for the protein GBF1, that it localizes to focal adhesions in glioblastoma cells. We still don't know what it's doing there, but there are lots of new questions from our lab to answer. And so one thing that I just want to point out, some of my more recent students weren't a part of this work, but this is why research is so exciting for students to get engaged with. There are always new and exciting questions to answer, and I really enjoy working with my research students. This is one of my favorite things about being at Birmingham Southern is working with you guys. So even though maybe you weren't a part of this story, thank all of you for the work that you've been doing in my lab as well. So that's it, and with that, I'll take questions. Oh, I'm sorry. So I'm really most interested in understanding sort of assemble and disassemble those focal adhesions. That means if we can block them, we can stop those cells from migrating by developing therapeutics. So that's what I'm really most interested in, is trying to understand what it's doing at the surface and if there are ways we can block that process in order to treat glioblastoma. That's probably our most exciting. We haven't looked that much, so we haven't seen it at the surface in any non-polarized cells. What I can tell you is that in Fission yeast, I don't know exactly where the guests are localizing, but we do see changes in the localization of some of the arcs that are localizing the tips. So we may also have this same directional phenomenon going on in yeast. So I'm also very interested in pursuing that question. I really like sort of living at the interface between the two. That's a really good question. So what is it? Is it a cause or is it an effect? And we don't know the answer, because one thing that may be happening is that if the right organelles, or if the lysosomes fuse too much or can't undergo fission, maybe you're not able to get the right proteins that perform those functions in or out. But it also could be that the sheer volume is diluting the concentration of the things that are in it. So it could go in either direction. And we, to this day, don't understand why the function is impaired.
You said that cells are bigger, vacuole, the whole cell is bigger. Do you have smaller vacuoles or the whole cell smaller?
So this is something I'm really interested in. So we think that there may be a link to cell size and vacuolar size, and there are some pathways that may be involved in. And I haven't done that work, but yes, there are other labs that have shown that if you affect cell signaling, it also affects vacuolar size. So yes, there seems to be a link between the two. And interestingly, if you starve cells, it affects cell size too. So if you starve them with glucose, the vacuoles get really big, and the cells get really big.